Half a day, and welcome to this video, which will be looking at the structure of RNA as well as some of the key differences between DNA and RNA. RNA stands for ribonucleic acid, and the monomer of a ribonucleic acid is called a nucleotide. So you may already see or hear some of the similarities between these two structures. Deoxyribonucleic acid, ribonucleic acid. Monomers of both of them are nucleotides. In looking at the RNA nucleotide, we see that it consists of three parts. Part 1 is the phosphate group, which we symbolize as a circle. Part 2 is the pentose sugar, which we symbolize as a pentagon shape. And part 3 is the nitrogenous base, which is usually symbolized as a rectangle. And these three parts make up our RNA nucleotide. Next, if we look at the RNA nucleotides, we're going to take a look at the nitrogenous bases. And again, with RNA, there are 4 types of nitrogenous bases. We have adenine, cytosine, guanine, and in this case, we have uracil. So if you recall, in DNA, we have thymine, but in RNA, thymine has been replaced by a structure, a very similar shaped structure, called uracil. And that was one key way to differentiate between the RNA nucleotides and the DNA nucleotides. Adenine will bond with uracil, cytosine and guanine still bond together. Uracil is also a pyrimidine in its chemical composition. If we look at the differences between DNA and RNA, first thing we'll notice is the sugar. Both are pentose sugars, which we can see here. We could label these 1, 2, 3, 4, 5 carbon. 1, 2, 3, 4, 5 carbon, so they're both pentose sugars. But we can look closer and we can see this one is deoxy, and we notice here that in the case of the ribose sugar, we have this OH, and here on this, the deoxyribose, it is without the oxygen, deoxy, so it just simply has the hydrogen on it and no oxygen present. So here we can see the difference in sugars between ribose sugar and deoxyribose sugar. If we look at the polymer structure of DNA and RNA, DNA is double-stranded and RNA is single-stranded. If we look at the nitrogenous bases in DNA versus RNA, DNA has adenine, thymine, cytosine, and guanine. Adenine and thymine join together, cytosine and guanine join together. In the case of RNA, however, the thymine is now going to be replaced by uracil. So we have adenine, uracil, cytosine, and guanine. So where there's an A, it will match up to U in RNA, and C and G will still be together. If we look at the structure of an RNA nucleotide, we can see that the carbons are going to be similarly labeled as DNA. This will be carbon 1, 2, 3, 4, and 5. So here we have the 5' end of the molecule, and this is the 3' end of the molecule. And this is very similar to the DNA structure. So this is the end of this video on the structure of RNA, and I hope you have a great day. Thank you.